produced an analysis and reported it on nearly 3,000 trial arms of treatments for COVID-19. These are registered trials on the big international registers. 95% of them were either too small or non-randomized, so only 1 in 20 trials had any chance of providing a useful answer to a relevant question. The rest were doomed to failure from the off. Second, those trials, and it's not just those trials, this is a broader statement, many trials, most trials, fail to recruit to time and target. Industry figures are typically that 85% of trials fail to recruit to time and target, and I would argue that academic trials or non-commercial trials are no better than that. So we've got 1 in 20 that is designed to succeed, 19 out of 20 does not. And what's that? About 1 in 6 that actually is designed to succeed will actually then recruit. We haven't really got halfway through the problem yet. The third element is the operational cost and complexity has spiraled out of all control. The contract research organisation has been growing exponentially. It was 30 billion in 2015, rising at least 10% per year, an exponential growth. There's nothing wrong with outsourcing, but when a model is rewarded on time and materials plus a margin, it rewards complexity rather than actually driving towards refining and simplifying and streamlining to get the right answer, efficiency. And finally, as a consequence of all of these things, even the successful trials are in the order of $1 to $2 billion. So $1 billion was about the cost of some of the big PCSK9 antibody trials from 4 or 5 years ago. I dread to think what the costs of the current generation of Alzheimer's disease trials. And remember, those are only in relatively niche populations. So what are the causes? I've touched on some of these. The first, I say, is around poor design. That's fundamental. The second is about cumbersome execution of the trials. There are plenty of manual, repetitive, duplicative processes. If you want a sort of measure of that, well, look at the fact that 40% of trial costs aren't for doing, they're for checking. 40% of the trial costs are going on checking that somebody else is doing the right thing, or also checking that the other people are checking, that other people are checking that somebody is doing the right thing. There is an underuse and poor use of routine healthcare data, whether that's for planning, how trials might be designed, where sites might be, where patients might, with relevant conditions might live, whether that's for enrollment or for follow-up of patients. There's a technological naivety. Plenty of people are inventing new apps which the patient can interact with, and that's good. But the back end is a collection of servers and systems that are like a 1980s or early 1990s server room. Or the equivalent of the Victorian sewer system on which the UK currently operates so poorly. They're also impractical. Every trial ends up with a patient, or a participant more accurately, and most trials end up with an interaction between a busy clinician and that participant, and yet there's little focus on what makes it easy and attractive for them. There is a regulatory rigidity. I don't mean about the results and the licensing decisions. I mean about the way in which trials are conducted, so that we have a set of good clinical practice rules, which were written largely in the early 1990s, if not before, and which are, if you like, predicated on the idea that carbon copy and the fax machine are state-of-the-art technology. And finally, we have these misaligned incentives, where complexity equals success, if you're in that industry. And the consequence is that many treatment decisions, as I said earlier, are based on inadequate information, that many novel treatments never make it because they're not worth $1 billion bet, or they are only developed for very limited indications. It's hard enough finding a medicine that works, and then to underuse it because the barrier to actually getting a proper evaluation in the scale of the population that has Alzheimer's, the full scale of the population that has heart disease, that's a tragedy, really. And many effective interventions are underused because their true impact is not understood. Think that it took 20, 25 years for statins to go from proof of concept in the 4S trial to really widespread use in primary prevention in kidney disease, in diabetes, in women, in the elderly, and so on and so forth. Think how long it took for the SGLT2 antagonists to go from a drug for diabetes to a drug that appears to be for substantial proportions of cardiovascular and renal disease. And fourthly, many ineffective or even harmful treatments continue to be used simply out of ignorance. We did a trial a couple of years ago, about 5 years ago now, 10 years ago, of niacin, a B vitamin, which had been widely recommended for the prevention of heart disease. When we did the trial in 25,000 people, it transpired that 1, it doesn't reduce the risk of heart disease. 2, that it increases the risk of hospitalization for serious bleeding and serious infection. A harm that had gone unrecognized for the best part of 4 decades, for a treatment that was second-line therapy in many of the international guidelines for prevention of cardiovascular disease, and for a treatment that specifically in the US had a billion-dollar market. And finally, the poor state of preparedness for future public health threats. For the next pandemic, we can't hope that the success of the recovery trial gets repeated. And that, I guess my very final point is we can't forget that not all people are the same. And not least, we need confidence that the trial results are relevant to different sorts of people. So with that in mind, in early 2020, February 2020, colleagues and I wrote this paper in the New England Journal of Medicine. We called it at the time Magic of Randomization versus the Myth of Real-World Evidence. That's a sort of terminology clash. If I wrote it again or retitled it, I would call it The Magic of Randomization in the Real World. It'd be lovely if you read the paper and cited it, but in reality, here is actually what it says to save you the time. Essentially, smart trial design, smart trial delivery, integrated with whatever routine healthcare looks like and whatever routine data systems look like, and they're not all the same, and supported by proportionate trial regulations and guidance, is the key to driving success for better patient care and public health. And on that note, I'll turn to my work of 2 weeks after that paper was published, which is the recovery trial. Here I want to take you back to the 9th of March 2020. On the 9th of March 2020, Jeremy Farrar and I, Jeremy Farrar is head of the Wellcome Trust, or at least was at the time, found ourselves on the same bus commuting between Marylebone Station in London to the Wellcome Trust building. And in the context of that 10-minute journey, we were discussing the outbreak, epidemic, I think it was just about by then declared a public health emergency, that was COVID-19. A mystery or previously unknown disease, very little known about many of its features, no known treatments. And of course, at that stage, it's all happening elsewhere. It happened in China and then it disappeared after about a month and we didn't hear any more of it again. It happened in Iran, it happened in northern Italy. That's getting a bit closer to home. But it actually then was starting to occur. The first cases were just emerging in the UK and of course in many other places around the world. And on that 10-minute bus journey, Jeremy and I were discussing the news we'd been having from colleagues in northern Italy, reflected in the serious newspapers on the left here, about the tragedy that was unfolding, of the hospitals being so full that they were having, and the ventilators being in such short supply, they were making very tough choices about who to put on a ventilator, and even tougher choices about taking people off ventilators. And the hospital car park was full, not of cars, but of mortuary vans. In British tradition, the more popular newspapers were dealing with the very British pandemic, which is there enough toilet roll to go round? That seemed to be the headline news. To Jeremy and I on that crowded bus, there was no doubt the tsunami was coming to the UK. The UK could look like Northern Italy in 2 weeks, and we had to get a trial up and running in 2 weeks. He had recently introduced me to Peter Horby, my colleague, who's an expert in infectious diseases, and that was our challenge for Peter and I. So what did we have at the time? What's our question? Well, this is a major public health crisis. 1 in 4 patients who admitted to hospital didn't make it out alive. It was worse if those people needed ventilators. Well, what treatments? Many candidate drugs, many opinions from all sorts of sources, not just medical, not just scientific, and even those that were medical or scientific weren't always well-grounded or well thought through. As it turned out, they weren't always right either. No reliable data, frankly, and unlikely, in our view, to be a single big win. This was not a disease like meningococcal meningitis, where a single dose of an effective antibiotic would be the solution. Much more likely that a series of moderate wins in a common problem, and then hopefully, if we can find a series of them used in combination, could have a big impact. And that's where you need randomization, and you need scale if you're really going to go for this. It was, of course, an unprecedented clinical challenge. The health service was overstretched. There were huge time pressures and personal stress from frontline medical staff, and there were large numbers of unwell, anxious, and often elderly patients. I could actually put that slide up today for the NHS in the UK, or for the US healthcare system, or for almost any other healthcare system around the world. We were all dealing with unsustainable challenges of disease as opposed to of health. But it's in that context we had to do a randomized trial. And so I look back in order to look forward to the trial that had actually got me interested in the whole area in the first place. This was published as I was in the middle of medical school, just heading towards the wards back in the late 1980s. The so-called Second International Study of Infarct Survival, or ISIS-2, named after the River Thames as it flows through Oxford. A famous trial for a one-page case report form for a factorial design to study 2 drugs for the price of 1 and get 3 answers. One works, the other works, both works. Focused on the outcome that mattered, mortality. And with this message on the front page of the protocol, the 12-page protocol. By far the most important determinant of the success of the trial is the extent to which the responsible physicians and nurses choose to enter their patients. Hence, the extra work must be and is absolutely minimal. Practical at the coalface is important. And so we designed the recovery trial. This is the sort of first iteration. Simple eligibility. What's the problem? Patients are going into hospital with COVID and we don't know what to do with them. What's the outcome that matters? 1 in 4 are dying. Mortality is the outcome that matters. Randomization to avoid the selection biases and pitfalls of observational data. And a very simple data collection process. 1-page consent form, 1-page case report form at the beginning and the end of the period, and then because we were now in the 2020s, we were able to link extensively to routine NHS data. We started off with a sort of parallel group design. Over time, as we built in confidence, we shifted to a much more efficient factorial design. We can talk about the subtleties of that design if we wish later on. As I say, routinely collected data from the NHS. Now, many people think, well, if there's one NHS, one logo and one funder, surely there's one data set. And the answer to that is, in your dreams, in my dreams, it's a complex environment. But we brought together something like 25 different national and regional data sets, covering all the elements that we could, tempered by all the data we could get hold of. Now, COVID can affect anyone. That was pretty obvious from the start. Of course, it's higher risk in higher risk individuals, the elderly, those with comorbidity and so on. So we had to make sure recovery was open to everyone. Every single acute hospital in the country, in the whole of the UK, took part. Some of the greatest recruitment actually happened in the so-called district general hospitals, not in the teaching hospitals. You know, for example, a hospital in the north of England, not a university hospital, recruited nearly 1,000 patients. Swindon recruited 500 patients. These are towns and cities that are not on the tourist map. These are towns and cities, hospitals that are not on the top of the academic league table, but which were able to deliver these trials at this scale Because they supported it, we made it easy for them. In the first 12 weeks, we randomized 12,000 people. We went from protocol on the 9th of March to first patient on the 19th of March to the first results on something like the 5th of June. We had a lull over the summer, and then, of course, there was a huge surge over the winter of 2021. We are still recruiting, but at very low numbers, as the tide of COVID seems to have gone out for a more sustained period. And, of course, we have vaccination, both of which are good things. So here we are on the 5th of June 2020, as I say, less than 100 days after starting the trial, with the first results, and they were for hydroxychloroquine. Despite the hope and hype that had gone before it, it turned out that giving hydroxychloroquine to people who are in hospital with severe COVID was of no benefit at all. Arguably, possibly slightly harmful, but we don't need to argue about that, certainly of no benefit. We announced the results the same day, and within 10 days, they were implemented across the world. In particular, in the FDA, where, of course, there was considerable political enthusiasm for hydroxychloroquine, the FDA issued this advice against the use of hydroxychloroquine within 10 days from that single result. And then we come to the 16th of June, the day that we announced the results for the steroid comparison for dexamethasone, a cheap and widely available drug. Costs about, in UK terms, about £5 for a course of 10 days. It's of similarly low cost all around the world. For patients on a mechanical ventilator, reduced the risk of death by a third. For patients who don't need a mechanical ventilator, but they might be on non-invasive ventilation, reduces the risk of death by a fifth. And for patients not requiring oxygen, there was no impact on death at all. So a really nice, clear result, not only overall, but actually recognizing that there are differences in the disease and the interaction between whether the treatment, how effective the treatment is or not at all, and the stage of disease, essentially the stage of the inflammatory response to the viral insult. We announced that result the same day, first of all, actually, to the science and health media in a press conference, because this was such an important health message, we wanted it to get out straight without any actual or perceived elaboration. Secondly, later that day, Peter went up and presented at number 10 at the nightly news briefings. This one result had an impact all around the world. Remember, this is at a time when we didn't have a vaccine. We didn't know for sure whether we'd get a vaccine, and we certainly didn't know when we would get a vaccine. So the first breakthrough treatment, if you like, that actually saves lives for people who would otherwise die from COVID. The NHS and the chief medical officers adopted that result within 4 hours. So here we have an example where a result is generated in less than 100 days. A trial is started in 9 days. A result is generated in less than 100 days. Less than 4 hours later, it's NHS policy across the whole country. By the weekend, it's saving lives. And internationally, within the following few weeks, was adopted worldwide. Clear results save lives. By the following March, the NHS put out a statement, which I at the time thought was a bit exaggerated, but I think in retrospect, certainly by now, I think it's probably about right, is that one result saved 1 million lives. We had people writing to the MHRA, to the chief medical officers, to ourselves and others, professors of medicine, saying, you shouldn't be trialling steroids. People who are fighting an infection do not need their immune system suppressed. It is dangerous to do so. It is dangerous to do this trial. They had valid reasons for constructing that hypothesis. Constructing a plausible hypothesis is no excuse for not testing a plausible hypothesis. And it was absolutely the case I didn't know whether hydroxychloroquine was going to work, steroids were going to work, or any of the treatments we've studied were going to work. Every one of them, I hoped would, not least because we had a pandemic to deal with. But it just shows the value of when there is uncertainty, when you have a hypothesis, test it and test it properly. And what testing it properly means is testing it to the point that you have convinced 90% of the audience. Think about what your audience is too. So we went on and studied a whole range of further treatments. And in total, we have found 4 effective treatments for high-risk patients in hospital, with 1 or 2 subtleties about that exact criteria between these results. And sometimes we get accused, well, you know, recovery, streamlined, simple, that's fine for repurposing old-fashioned drugs that we know lots about and have been around for decades. This is fine for academia, this will never pass regulatory muster, etc., etc. 1 million lives saved. Dexamethasone, yes, old-fashioned, well-known, widely available. Tocilizumab and baricitinib, most people outside rheumatology and 1 or 2 other specialist conditions are not familiar with those drugs. Many people, including the British Prime Minister at the time, can't even say the names of some of those drugs. And yet what we were taking here was licensed, patented drugs and repurposing them, label extension if you like, but it's not exactly a linear extension from rheumatology into COVID, and demonstrating additional value on top of the steroids of further suppression of the immune system. And then finally, in the bottom right-hand corner, the monoclonal antibodies produced by Regeneron in this instance, demonstrating that in an entirely new drug, it didn't exist on the day we started the trial. In an entirely new and unlicensed drug, one can get convincing results about the role of monoclonal antibodies in patients who are in hospital. The views had switched. Is it too late now to tackle the virus? No, it turns out it's not. In those people who are not mounting an antibody response of themselves, giving them antibodies will reduce the risk of death. If they are mounting an antibody response themselves, giving them more antibodies is expensive and useless, a bit like giving vitamins to people who are already vitamin replete. So if those are the positive results, what about the so-called negative results? None of these are negative. Every one of these had big impact. These are all widely recommended, loudly promoted, extensively used in the guidelines, resource-intensive in some cases, like convalescent plasma, and it turns out in a hospital setting, useless. We have had, I think, 1 or 2 further more than this, but I couldn't fit them on the slide. So how did the regulators view this? So here is a review piece, or a viewpoint piece, published in JAMA Internal Medicine at the end of last year, from the 3 most senior figures at the FDA, which focuses on recovery trial, but concludes streamlining and quality are not opposed. People have often said to me, well, you're cutting corners. What about patient safety? This is all very well for well-established drugs, not for new drugs. Streamlining and quality are not opposed. And so I want to finish this section just by recognizing that having talked about, if you like, the therapeutic, the clinical pharmacology, the therapeutic, the trial methodology, and so on, having talked about the impact on public health and on regulatory decision-making, we now just remember that what we actually do this for, and the only way we can do this, is with the patients. And this gentleman, in fact, is a local from here. He's a Glaswegian man and his wife, and this is her quote: "When he left in the ambulance, I really didn't think I would see him again." Now, he's gone on to give a number of interviews since he took part in the trial, explaining just how important it was to them to help find solutions. And many other patients have done likewise. 48,000 patients have now taken part in the recovery trial. It would be impossible without them. Sadly, many of them are not with us, but we are grateful to them all, as are future patients. So I'll conclude this section just by thinking about recovery in numbers. So it was fast, 9 days to the first participant, 99 days to the first positive result, 4 hours to policy change. It was efficient, It took 25 days to get to 1,000 patients, about twice that to get to 10,000 patients, 9 months to get to 20,000, 20 months to get to 40,000 before the pandemic thankfully subsided and vaccines started to play their role. It was representative. This was 1 in 10 of all UK COVID admissions took part in the trial. The proportion who are from non-white ethnic groups is exactly the same as the proportion in those patients who were coming to hospital with COVID. Likewise for women, including 100 pregnant ladies. We did not exclude pregnant ladies. Obviously, there was some modification for specific drugs. 1 in 5 of the patients were aged over 80. And we also recruited 300 children, not only with the respiratory form of COVID, but also the inflammatory form. I'd argue that it was productive. We have 13 results to date. I've shown you some of those. There are 4 ongoing. We've studied repurposed generics, label extension, novel agents at a total cost of less than £20 million. In round terms, it is roughly speaking £40 per patient per answer. Trials do not have to be hard, they do not have to be expensive, they do not have to be exclusive. They can produce robust results at low cost. And it's certainly been impactful. I've shown you some of that, but also just to highlight, not only impactful in terms of the context of a pandemic, emergency use authorizations, guidelines, and the rest, but actually impactful in terms of real licenses. So the tocilizumab, or Actemra as it's branded, I believe, licensed in 35 regions on the basis of the recovery results, including by the FDA, accepting, probably for the first time, results from an academically led platform trial using routine real-world health data in the middle of a pandemic and using that to actually change licenses for drugs. And it's beneficial in the sense that we've found 4 effective treatments and saved so many lives. So I'll go back to my thesis, which was around reinventing randomized controlled trials. And I think I've probably given you a useful sort of exemplar, shall we call it, a case study, almost a prototype, perhaps, in the recovery trial. And picking up on these themes of smart design and delivery, integration with routine healthcare and data systems, and supported by proportionate trial regulations and guidance. So as we look forwards, what do we need? And I guess this is in part, well, what do we need from whom? And a lot of what I would say here is that this is what, if you like, government or the system or policymakers, the people out there could be doing to make clinical trials easier and better. The first is to establish and support the trials infrastructure over a very long period of time. This would have been impossible if Peter Horby hadn't been supported for 20 years in studying emerging infectious diseases. If I hadn't been supported for 20 years doing clinical trials in cardiovascular and renal disease. If the programmers, the trial managers, the data managers, the people who submit ethics applications, the people who manage drug supply, all those things hadn't been supported over a very long time. Not just in academia, but also the investments that were made in various parts of government, in the National Institute for Health Research's research nurse system, and so on and so forth. We need sustainable investment in people skills, partnerships, data and technology, but also in policy and processes. The second thing is we really need to build on this theme of integration with the health service. This is not the same as saying every hospital doctor should be doing clinical trials, or that every patient must be in a clinical trial. I mean, that's clearly nonsensical. Patients have the choice about whether they wish to be in clinical trials, and long may that remain the case. But this is about thinking about, within the health journey, thinking about research and clinical trials specifically as being a component of what makes a good healthcare system, a good hospital, a good primary care practice, a good public health system, a good doctor, is, I think, a fundamental shift that's been necessary. So often we see audits, we see guidelines, we see policy. Rarely do we see it backed by evidence. If you want a figure on that, the big cardiovascular guidelines produced by the American Heart Association, American Congress of Cardiology, the European Society of Cardiology, and so on, 85% of the recommendations in those guidelines are not based on sound evidence from randomized trials. It's not my data, that's Rob Califf's data, and he's now FDA Commissioner. And he studied that first in 2009, and 10 years later in 2019, and the number was the same. So cardiology sits at the top of the evidence-based tree. Heaven help the rest of medicine. So we cannot just muddle on in uncertainty, but confidence confident uncertainty. So we're confident that we know what we're doing, whereas in reality, we just don't have the evidence. Don't guess, randomize. That was the theme at the beginning of recovery, and that remains the theme in so many other areas of medicine. We have to optimize the use of data. Now, people who are into data will say, well, what about the FAIR principles and so on and so forth? Yes. So the principles of using healthcare data for trials are much the same as they are elsewhere. But the things that are special, if the data are not accessible, by and large, primary care data in the UK is not accessible. Getting anything in from the depth of the hospital record systems is a challenge. They're not analysable. If they're not interpretable because you don't really understand what the data items mean, and if they're not acceptable to regulators and policymakers, decision makers, then we have a problem. And of course, one of the things that the regulators did focus on in recovery was, you've used all this real world data in your randomized trial. That sounds scary. How do we know it's right? They seem to have ignored the fact that all their pharmacovigilance activities rely on exactly those same data. They're making decisions all the time about the safety of medicines based on what they call real-world data. It's the same data, only we've inserted randomization, so we have a sporting chance of actually getting the answer right. We need to think about collaboration across organizational boundaries and with patients and public, and we need to think of regulation quite differently. In my view, and remember I am a clinical pharmacologist, just like most of you, in my view, the main aim of a national or international drug regulator is to make sound decisions about the safety and efficacy of individual drugs and treatments and strategies. Make sound decisions in order to improve and protect the health of the patients in their, let's call it, jurisdiction. Those decisions are so much easier if you've got good evidence from trials. So we need to refocus regulation that it enables good trials to happen, rather than simply seeing them as risky and somehow that the 85% of clinical guidelines that are based on no evidence, no good evidence at all, is somehow not risky. We have to reshape this. We need to emphasize the principles and not the processes. Think about flexibility and innovation, and focus on what actually makes for high quality, rather than simply what documents have you piled up. To give you a sort of snippet on that, both the recovery trial and many of the cardiovascular and renal trials I've been involved in have been audited and inspected over a number of years. Not once has an inspector said, can you show me the randomization system? Not once has somebody said, I want to see how that randomization system performed. And yet, that is the one thing that is critical to quality. They've checked training certificates, they've checked GCP certificates, they've checked source data, and all this other stuff. They've never checked the thing that matters. So what can we do in data? Well, this is sort of highlights of work that colleagues have done in Oxford, and I've been involved in sort of more peripherally. But we can use data to drive planning. So the top left-hand slide, planning for where do we establish the clinical trial sites in that instance. On the bottom left, we can do that for recruitment. So use the routine healthcare data to invite over 1,000 patients who are known to have, by the NHS Scotland in this case, to have diabetic retinopathy, and invite them to take part in a trial. Those dots now are not sites, those are people's houses, that's where they live. The bits which have large gaps in them are because there's not many people and many deer, and the deer, as far as we know, don't get diabetic retinopathy. At least they don't respond to the letters. We can also use data if we have the data constructed properly and the right technology for things like quality control, for statistical monitoring. In this example, the further north you are, further up the y-axis, the more extreme the site performance is on a number of metrics. The site size is the size of the blob. And you can see that in that top right-hand corner, there are 4 sites that look unusual, exceptional, and probably not for a good reason. Why do we do trial monitoring going to those yellow sites, which are all much of a muchness, on a repeated 6-week, 3-month, whatever it is, basis in checking source data? Why don't we actually focus our resource on those 4 at the top right-hand corner, which are clearly doing something odd? And finally, we can use data, the hospital episode statistics, to assess clinical outcomes. This is an analysis done by colleagues and one of my PhD students, looking at the results of the ASCEND trial of aspirin and of fish oils. In the trial, aspirin worked and fish oils didn't. But the black lines are the results you get from the carefully curated, adjudicated, etc. data. They are the trial results. In the grey lines are what happens if you simply go to the NHS and say, can you tell us who's been into hospital, who hasn't been in hospital, and what the diagnosis is? You get essentially the same answer. Your data don't need to be perfect, particularly in the context of randomization, to be useful. We also need to think about regulation. Here's work that's been funded by the Gates and the Wellcome Trust I've been involved in around what actually does a good trial look like. Whether you're a funder, whether you're a regulator, whether you're a GCP inspector, whether you're a participant, whether you're an investigator. What are the things you really need to focus on? Well, first of all, get the right answer. Informative and relevant. Without that, everything else is a waste of time and worse. Respectful, obviously, of the participants, collaborative, transparent, feasible, and of course you do have to manage the things that matter, the quality of those well. And those thoughts are fed through to a number of national and international efforts. So here's the G7's 100 Days mission published a couple of years ago. And in that, they pick out clinical trial regulation as being one of the things we need to do now in advance of the next public health emergency if we want to have a good chance of being able to respond to that public health emergency. And as I've explained to you, also if we want to tackle the many common diseases that are actually causing such a challenge at the moment. Refocusing on the guidelines on the fundamental scientific ethical principles that underpin trials, embracing flexibility in innovation, and focusing on what matters for the generation of actionable information rather than just what is easy to check, placing an emphasis on principles and purpose rather than process. So that was the G7 2 years ago. The ICH, so the International Club of Regulators, I'm not sure that's exactly what it stands for, but that's essentially what it is, have just, in the last month or so, issued new draft guidance for comment. And I really strongly encourage you to read it carefully and read it with two perspectives in mind. One is, will this help me to do a better trial? And secondly, what in here is at risk of overinterpretation by somebody else, by a policeman, inspector, the quality person from some company, that would actually make it harder to do a good trial? But there are some very strong themes in here, to be clear. Trials with inadequate design and or poorly conducted waste resources and efforts and time of investigators and participants. The principles are designed to be flexible, encourages thoughtful consideration. Careful considerations of the factors that are relevant to ensuring trial quality is needed, and so on and so forth. So I say that consultation's open right now. I encourage you to respond. And the World Health Organization has a piece of work kicked off at the World Health Assembly last year. Again, picking up on that G7 sort of recommendation, there needs to be efficient governance, focusing on the fundamental scientific and ethical principles, acting proportionally, and embracing flexibility and innovation. These themes are beginning to come through, but it's so critical that they're not just nice words in policy papers. They're not even in the details of procedures. They are actually embodied in the actions of people on the ground across all organizations. So 2 slides and I will finish. So I think that, to conclude, the arbitrary use of unproven treatments is damaging to patient care, it's damaging to public health, and COVID illustrated that, but actually there have been other examples too. And randomized trials are really a critical component of what it means to have a high-quality clinical care system as individual doctors and nurses and healthcare professionals, but also as a NHS in this country or more broadly. Compelling results change practice. We did have a statistician go over the results for the recovery trial on dexamethasone repeatedly. In fact, we had 2 statisticians going over them repeatedly because the results were so impressive. We wanted to make really sure they were right before announcing them to the world. But in a sense, once they were presented, you didn't need a statistician to know what they meant. That's what I mean by compelling results. And they did change practice in that instance within 4 hours. And so regulatory, policy, clinical decision-making is much better and frankly much easier when the evidence is clear. There is no need for pontification and endless committees when the evidence is clear. So let's focus on what's needed to get the evidence clear, as opposed to trying to find workarounds and prevaricate or avoid the issue. For success, trials must be designed to deliver actionable results, and they must be feasible for patients and clinical staff. Anybody can design a trial that nobody can do. The trick is to design a trial that everybody relevant can do. And on that note, obstacles, burdens to randomized trials, they damage patient care. They damage and are harmful to medical innovation and the opportunities that are coming from the biological and technical revolutions of the last couple of decades. They damage public health. And those obstacles and burdens damage and destroy the trustworthiness of the health system. We have to get this right. You mentioned protests very kindly at the beginning in the introduction, and just to give you a sense that this is what I'm doing next. So this is my contribution. Please make yours. We established a not-for-profit company, an organization, to really think about how we could play those same lessons into many other common and life-threatening conditions, from Alzheimer's disease to common infections, common cancers, and so on. Whether that's using brand new drugs, entirely novel technologies, or repurposing, and so on and so forth. And really focusing on, yes, the design, number one, and the delivery, thinking about how we use data and technology, thinking about how we work with players across the whole system, from industry, I guess, and government at one end, through the healthcare system, through clinicians, out to patients and public at the other end. And really thinking about optimizing policy and regulation in 2 regards. One is establishing the need for trials, the centrality to good healthcare that trials play. And the second is actually improving the quality of trials in a genuine way, as opposed to merely a data checking exercise. And with that, I'll stop, and thank you very much.
Thank you very much, Martin, for a fantastic talk. Open for questions. Can I start off with dexamethasone? I think we discussed this during the pandemic. How did you come to 6 milligram choice? And I think you were doing further trials with a higher dose. And where have you got to with that?
Yeah, thanks very much. That's a very good question. So the first thing is that, I'll go one stage back, the choice of drugs overall. Initially, or quite soon, we realized that the breadth of choice and the amount of expertise needed was beyond Peter and I. And so very grateful for Munir and others for sitting on the UK COVID Therapeutics Advisory Panel, which we asked to have set up to actually provide us with that expert advice. The choice of drugs, there had to be some reason for believing it might work. It could be from animal data, lab data, it could be from use in other contexts. We had to know something about the safety. There had to be enough drug to actually study in several thousand people. And that ruled out a whole bunch of complement inhibitors and whatever else. And there had to be the potential to have enough drug to treat many hundreds of thousands of patients, given the scale of the pandemic. The specific choice of dexamethasone dose was based on some previous work that people had done in influenza, and I think there was a case of not too high and not too low. And finally, that choice I think is right, because we did study higher doses. If suppressing the immune system is good, could we increase the dose of steroid? And in people who are on oxygen but not on non-invasive ventilation or ventilator, so they only need oxygen, then in fact increasing the dose of steroids is harmful. So that was our first result, where we actually demonstrated really clear harm, and in fact the DMC asked us to stop that part. But we continue to study what the situation is in people with the more severe disease, people who need a non-invasive or invasive ventilation.
Great, thank you. So I think, Jeff, you were first, so you go ahead.
Thanks, Jeff Ardenson from Oxford. Thanks, Martin. That was clear, sometimes depressing, but constantly on the ball and spot on. Thank you very much. You mentioned the important point about testing hypotheses. People often say, I'm doing this to show that X does Y, instead of to test the hypothesis that it does it, or to test the null hypothesis that it doesn't do it. Now, you showed many graphs from the recovery trial where the lines were on top of each other, no effect, or the intervention below, effect, excellent. There were two cases where it was above the line: hydroxychloroquine, dexamethasone, mild cases. And you presented them as no effect, but the question is, are they harmful? Does hydroxychloroquine cause arrhythmias, interactions, and so on? What were the QT intervals? And is it wrong to use a steroid in mild cases because of immunosuppression, whereas in severe cases you need immunosuppression? But underlying that, those two specific questions, is a more general question illustrated by your niacin study, and that is the problem of detecting adverse reactions in clinical trials when you need so many more patients than you do to show benefit, and determining the benefit-harm balance can be lost.
You've put a lot of questions in there, Jeff. So there's no point in using a treatment that doesn't work. So in a sense beyond that, whether it's harmful or not becomes to some extent an academic question, in the sense that why expose somebody to a drug that is not going to help them? And that then dismisses, then if they don't get the drug, they'll certainly never get the harm, even when it exists. On the hydroxychloroquine, if you looked at the other aspects, so the arrhythmias and so on, there wasn't any clear signal for that. The steroid in low-risk people, I mean, what we didn't study is the lowest risk people, in other words, people in the community, and there I would really worry that actually that might be genuinely harmful, but there hasn't been a good trial in that context. And finally, I think that when I talk about compelling results, I'm not after testing simply the hypothesis, does this have benefit on the thing I think it's going to have benefit on? I'm actually interested in testing the hypothesis, testing more than that. Is this drug going to be useful? Which is why I think when one asks the question, one has to think about the audiences. One audience is, does it get into the New England Journal or the Lancet? That's a p-value question on the primary endpoint. The second question is, does it get licensed? That's also much the same. But actually, should it be used and so on becomes a broader question. And I think when one's doing the types of trials in the types of context I am, you have to think about the end game. And the rest will actually fall into place, because you will naturally have to answer all the earlier players' questions.
Thanks. So there's 4 more people waiting, and we are running out of time, so please, short questions and short answers. So Michael, please.
My name is Michael Edelston. I'm in Edinburgh, clinical toxicologist, so I do trials of antidotes. Just a couple of points is, the MHRA uses your recovery trial protocol as an example of a short, brief, to the point, and they praise it. But when I write my protocols within Edinburgh, the systems within Edinburgh continuously extended, and every time I go back, there's new sections that have to be filled in. So somehow there needs to be guidance from the MHRA to the universities or to the sponsors to say, we really don't need to have such extensive, every single possible imaginable problem. So that was one point. The second one is...
On that I agree, but I'd also say that not everybody in the MHRA understands that point.
Okay. Kirsty Withenshaw does.
She's left.
Oh, right, okay. She was very positive about your protocol. The second point is, what you don't mention is that you know, the trial is absolutely brilliant, amazing. It changed, it transformed the world. But it benefited from a huge system in place across the UK of clinical trials, which were all stopped. So my trials were all stopped, and we switched to recovery almost overnight. So I'm not sure whether your £20 million cost for all this beautiful work actually includes what was already in place, and fundamentally what just changed for at least 6 months or a year was all on recovery and on the vaccine trials. So do you include that in your 20 million? Because without that, you would never have been able to recruit so many so fast.
I include all the costs of the research nurses and so on, so the local support costs in that 20 million. The 20 million is not my grant. The 20 million is the total cost that was put in, including research nurses and so on. But I accept that there are wider knock-ons. We can talk about what the judgment decision. I'd also say, as I put on my slide, that getting sustainable infrastructure is key to this.
But they were there in place.
Michael, sorry, I better let other people have a go. Mike Spenning.
I'm Secretary General of UFOL, and I'm desperate issues about resource-limited countries which are completely different problems and need completely different simple protocols where there are clinical pharmacologists, but without the background support systems. And the question is, can we do anything without the background support systems with intelligent designs?
Yes, I mean, so in recovery we did have half a dozen lower income countries. On the high dose steroids, Nepal put in half the patients and their results were the same as the UK's essentially.
Great.
But again, we need sustainable infrastructure. That's why I included the WHO piece in this. You know, this is a global problem.
So microphone 3 and then microphone 2.
Morning, thank you for the presentation. My question is just based on if you have any data relating to high-dose low molecular weight heparin. The reason is because you mentioned about aspirin, and we also know about COVID-related deaths, which is due to thrombotic nature in some cases.
We didn't study heparin. There were a couple of other smaller trials did study heparin, and the results are slightly complex, so I'm not going to say them now because I'll get them wrong. In my view, it wasn't studied early enough, as in early enough before people had set about their protocols. So they established what standard of care was without evidence, which then made it hard to get evidence. So I think it's a semi-answered question. It's just unsatisfactory.
Thank you.
Please.
Hi, lovely overview, Matthew Duke from New Zealand. I think some lessons for us in terms of as our health system changes for the data. But my question is really about how to deal with the changing face of disease. So this is a disease that was evolving over the time at the same time as you're implementing treatment.
Yes, I mean, the most obvious example of that is the monoclonal antibodies. And that result that I showed for the Regeneron antibody would not occur now because it's not an effective antibody. In a sense, you don't need to repeat the trial to know that, but you can work that out in the lab. I think it's established a principle about antibodies being effective. I think the other things that have changed is things like age profile and the extent of inflammatory disease and so on. I think I'd argue that by the time people get to needing oxygen, then certainly onto needing other forms of respiratory support, it's sort of not a question of how you got there, you know, which variant got you there and so on and so forth, whether you're vaccinated or not, you are there with that. And I suspect that the clinical picture is very similar at that point to what it was 2 years ago. And at that point, then the steroids and the tocilizumab will be effective. But the number of people who are getting there is thankfully, the proportion of people who are getting there is thankfully much lower, largely due to vaccination.
Great. Thank you very much, Martin, for a great talk. Thank you. So I think it's coffee time now before the next session in this room.